So let us discuss on acid-base balance. An important part of being healthy is for the blood to maintain a normal pH range, that is the degree of acidity or alkalinity. So the normal pH of arterial blood is 7.4, while the pH of venous blood and the interstitial fluid is 7.35. The pH of blood is maintained between the 7.35 to 7.45. And all the activities of the enzyme system in the body are influenced by the hydrogen ion concentration. That is an indicator of pH. So changes in the higher hydrogen ion concentration alter all the body and cell functions, conformation of biological structural component, uptake and release of oxygen. So these are the effects happen if there is a deviation in the pH or changes in the hydrogen ion concentration. Now, what are the metabolic sources of acids which alter the blood pH? So there are two types of acids are being generated in the body. One are the fixed acid, another one are the volatile acids. So fixed acid or the non-volatile acids are phosphoric acid, sulfuric acid, pyruvic acid, lactic acid, and keto acid. While the volatile acids we can breathe out through the lungs. So that is the carbonic acid. Now, metabolic sources of bases. So they can be the citrate salt of fruit juices. They may produce the bicarbonate salt. It's not the citric acid. Citric acid is an acid, but if it is the salt of sodium, so citrate is a conjugate anion with conjugate base. So this conjugate base and sodium combination, so they may produce bicarbonate when they meet with water. So deamination of amino acid produces ammonia. That is another source of metabolic bases. Then formation of bisphosphate also contributes to the alkalizing effect. So these are the three major important sources of bases in the body. Then what are the regulatory mechanism to maintain the normal blood pH? So there are three important mechanism operating in the body all time to maintain the pH of the blood. One important is the buffer mechanism. That is the first line of defense. Then respiratory mechanism, that is the second line of defense. Then the renal mechanism, that is the third line of defense. So these three line of defense, they work to maintain the normal blood pH. Now, what is a buffer? Buffer is a substance that can resist the change in the pH even after addition of strong acid or bases. So it can be a mixture of weak acid and its salt of its conjugate base. Like we can give the example of like sodium carbonate, that is a salt of carbonic acid with sodium, and the carbonic acid is the weak acid. So this combination is a buffer. Then if one molecule differs from another by only one proton, the two are called the conjugate acid-base pair. So suppose in the water there is a carbonic acid and this sodium salt is broken up into sodium plus and bicarbonate ion. So this bicarbonate is a conjugate base of this carbonic acid. This is what they want to say here also. This is the bicarbonic acid. So in water, it is dissociates into H plus HCO3 ions. So HCO3 is the conjugate base of carbonic acid. It is differ by one proton, right? So buffer can reversibly bind hydrogen ions, and the general form of buffering reaction is the buffer system do not eliminate hydrogen ions from the body or add them to the body, but only keep them tied up until the balance can be re-established. So overall, in nutshell, the concept of buffer is like that. If there's an excess of H plus ion concentration, buffer catches it and maintains the pH. If there is a deficiency of hydrogen ion in the blood, so this buffer releases the H plus ion and maintain the pH. So this is the mechanism of buffer, how it works. Now, buffering capacity depends on concentration of the buffer, then relationship between the pKa of the buffer and the desired pH. pKa is basically it is the ionization capability of any acid. or any buffer. And pH means it is the negative logarithm of the hydrogen ion concentration. So a buffer has the maximum buffering capacity when its pK is very close to pH, right? For the maximum blood buffering capacity, the pK of the buffer should therefore be near 7.4. For example, the histidine in the proteins, they maximally contributing to the buffering capacity because its the pK value is 6.1, which is very close to the pH of the blood. So that's how it participate maximally in the maintaining the pH of the blood. Now, what are the blood buffers? There are blood buffers. There are three buffer system. One is the bicarbonate buffer, one is the phosphate buffer, and one is the protein buffer. So, in the extracellular fluid, the buffers are mainly this carbonic acid and bicarbonate ion system, then sodium hydrogen phosphate and sodium dihydrogen phosphate system, then sodium protein and hydrogen protein system. So these three systems are operating in the extracellular buffer fluid and working acting as a buffer. Then intercellular fluid buffers, they are potassium hydrogen carbonate and carbonic acid, then dipotassium hydrogen phosphate and potassium dihydrogen phosphate system, then potassium hemoglobin and hydrogen hemoglobin system, then potassium hemoglobin oxyhemoglobin system, and HHBO2 system. So these are the three systems operating in the blood. Now let's discuss about the most important buffer, that is the bicarbonate and carbonic acid buffer, how it works, how it maintains to the pH of the blood. So bicarbonate buffer system is the most important extracellular buffer. It plays an important role in maintaining the blood pH because of its high concentration. So the two elements of buffer, the one is the bicarbonate ion, that is a conjugate base, and the carbonic acid. They are regulated by the kidneys and by the lungs respectively. So mechanism of action, we will see how this buffer works. So when the strong acid like HCl is added to the bicarbonate buffer solution, so the increased hydrogen ions are being buffered by bicarbonate. They are taken up by bicarbonate to form the very weak acid, carbonic acid, which in turn forms the carbon dioxide and water. So you can see here in this particular equation, so if there is an excess of hydrogen ion concentration, like patient is in a acidotic state, right, metabolic acidotic state, so at that time hydrogen ion concentration in the blood is very much, so that is to be scavenged. So this bicarbonate buffer, this bicarbonate base, it is going to combine with this H plus ion and it converts up into the carbonic acid. Then finally carbonic acid is converted up into carbon dioxide and water in the lungs, and the carbon dioxide is being thrown out. So this is the way of scavenging H plus ion and relieved of carbon dioxide. When the sodium hydroxide is added to the bicarbonate buffer, so what will happen in that situation? Suppose this is the sodium hydroxide and there is a carbonic acid. So how it maintains the pH? The sodium hydroxide combined with the carbonic acid to form the sodium hydrogen carbonate and forms the as water. So alkaline effect, the effect of this NaOH is being compensated by the formation of this sodium salt of this carbonic acid, right? So any non-volatile acid stronger than carbonic acid can be buffered by bicarbonate. Then plasma bicarbonate is a measure of base that remains after all the acids stronger than carbonic acids have been neutralized. It represents the reserve of alkali available for neutralization of such a strong acid, and it has been termed as the alkali reserve. So if you remember the Anderson-Hasselbalch equation, we have seen the bicarbonate concentration is always 20, nearly 20 times higher than the carbonic acid. So that is alkali reserve. So all time acid concentration, because acids, there are a lot of sources of acid production in the body in various metabolic reactions. We have seen so many metabolic acids are being produced, like lactic acid, organic acid, so many acids are there, so they need to be neutralized. So always the this alkali or bicarbonate concentration is always higher, at higher site, in order to compensate the excessive production of acids. So that's how this is known as alkali reserve. Now at pH 7.4, the average normal ratio of the concentration of bicarbonate and the carbonic acid in the plasma is 25 millimole to 1.25 millimoles. So that is equal to 20:1, as I have already told you. Then subsequently, any changes in the concentration of either bicarbonate or carbonic acid, and therefore in the ratio bicarbonate and carbonic acid, is accompanied by a change in pH. So the two elements of buffer system, the bicarbonate, that is a conjugate base of this carbonic acid, they are regulated by increasing or decreasing the rate of reabsorption of bicarbonate by the kidneys. So if there is an excessive of bicarbonate, so they has to be removed through the kidneys, washed out through the kidneys. Or if there is a deficiency of this bicarbonate, so alkali has to be conserved, so that is being reabsorbed by the tubules of kidneys. So by altering the rates of removal or retention of bicarbonates by the lungs, this is the second way. So as I've already explained this, by altering the rates of removal of or retention of bicarbonate. So if there is an excess of H plus ion in the cells in the bloody fluid, so H plus combined with bicarbonate, it forms the carbonic acid. It goes to the lungs, where it is being broken up into the water and carbon dioxide is being washed out. So this is the one mechanism. Now, mechanism of action of phosphate buffer, how it works. So when a strong acid such as HCl is added to the phosphate buffer, the H plus is accepted by the base, conjugate base of this phosphoric acid. So that is this HPO4 minus. So it is being, the H plus ion is being accepted by the base, that is HPO4 minus, and is converted to H2PO4 minus. And the strong acid like HCl is replaced by a weak acid, right? So this is how it maintains the pH of the blood. When a strong base such as the NaOH is added to the phosphate buffer, then OH is buffered by the H2PO4 to form the HPO4 and the water. Thus, the strong base NaOH is replaced by the weak base HPO4. So this is another way how to compensate the effect of excessive hydroxy ions. So at the plasma pH of 7.4, the ratio HPO4 minus to H2PO4 minus is 4:1. So this conjugate base is always in the higher concentration as compared to its conjugate acid. For this system, this is comparatively acidic to this conjugate base. So organic phosphates in the form of 2,3-phosphoglycerates, that is 2,3-BPG, present in the erythrocytes, accounts for about 16% of the non-carbohydrate buffer of the erythrocyte fluids. Now, protein buffers, how do they work? So, like sodium protein and hydrogen protein system. So, in the blood, the plasma proteins, especially the albumin, act as a buffer. And albumin this consists of so many amino acids. So, buffering capacity of the albumin is because of its constituent amino acids. So, in the acidic solution, the basic amino group takes up the excess H plus ion and forming the this kind of ammonium ion. Whereas in the basic solution, the acidic component, acidic this group, that is the COH group, gives up the hydrogen ion, forming the OH minus of the alkali to water. So this is how they maintain the pH of the blood. Other important buffer group of protein in the physiological pH range are imidazole group of histidine. So each albumin molecule contains 16 histidine residue. And histidine, like imidazole group in the histidine is very, very important because of presence of this imidazole group, and it has got a labile hydrogen. So histidine pK value is very close to the pH of the world, so the blood. So that's how histidine maximally contribute to the buffering capacity of the blood. So that's how the concentration of this histidine residue is higher in the albumin. Then hemoglobin buffer. So this is potassium associated hemoglobin and hydrogen associated hemoglobin. So this is KHBO2 and this is the oxyhemoglobin, which is the potassium associated with potassium, and this is the oxyhemoglobin associated with H plus ion. So hemoglobin is the major intercellular buffer of the blood, which is present in the erythrocyte. And each hemoglobin molecule contains 38 molecules of histidine. Again, histidine is in the maximum concentration as compared to the other amino acid. The imidazole group of histidine has a pK of approximately very close to the this blood pH. So it buffers the carbonic acid. The action of hemoglobin buffer. Hemoglobin works effectively in a non-incorporation with bicarbonate system. So we can see here. So try to understand. This is the lung, this is the blood, and these are the tissues. So in the tissues, as a consequence of a lot of metabolism, there is a production of H plus ions, or the carbon dioxide being produced, or and this associates with water to generate the carbonic acid, which is broken up into H plus ion and bicarbonate ion. So then this H plus ion is being taken up by oxyhemoglobin, and it forms a temporary complex, that is the this is hydrogenated hemoglobin, and in this course of formation, there is a release of oxygen, and this effect is known as Bohr effect. And because of presence of this H plus ion, it facilitates the downloading of oxygen into the tissues, right? Like in the RBCs. Then this hydrogenated hemoglobin via blood, it reaches to the lungs. In the lungs, what happened? This hydrogenated hemoglobin now combines with oxygen. So it reverted or converted back into the oxyhemoglobin, and it releases the H plus ion, just opposite to the phenomena which is happening in the tissue. So the H plus ions are now released, and this H plus ion, here also the carbonate system is available because H plus ions are there. So counter ions are bicarbonate ion, and this bicarbonate abundantly reach to the lung side, and in exchange the chloride is shift to the tissues via blood. So this is, and this bicarbonate is now generating the carbonic acid, and this carbonic acid is being broken up and releases the carbon dioxide and water, and through expired air this carbon dioxide is released into the atmosphere. So this is how this whole mechanism is all this, the respiratory mechanism of transportation of oxygen and carbon dioxide via hemoglobin via bicarbonate system. So this is facilitating, this all is facilitating, this buffering capacity is facilitating the phenomena of respiration. So the transport of an appreciable quantity of carbon dioxide released from the tissues without change in pH is called the isohydric transport of carbon dioxide. So this kind of transportation uh, where the appreciable quantity of carbon dioxide is released from the tissues without changes in pH is called as isohydric transport of carbon dioxide. Then most of the carbon dioxide is transported in the plasma as bicarbonate, and because this bicarbonate is much more soluble in blood plasma than carbon dioxide, so this indirect route increases the blood's capacity to carry carbon dioxide from the tissues to the lungs. So this is the advantage of this process, how the carbon dioxide is transported in the form of bicarbonate. So then second line of defense mechanism, that is the respiratory mechanism. So it against the acid-base disturbances. So it functions by regulating the concentration of carbonic acid in the blood and other body fluids by the lungs. So respiratory centers regulate the removal or retention of carbon dioxide and thereby carbonic acid from the extracellular fluids by the lungs. So increase in H plus and and the carbonic acid stimulates the respiratory centers to increase the rate of respiratory ventilation, and excess acid in the form of carbon dioxide is quickly removed. So any increase in hydroxy group or the bicarbonate depresses the respiratory ventilation, or that means the alkalinity in the blood will depresses the respiratory ventilation and release of carbon dioxide from the blood. So the increased blood carbon dioxide will result in the formation of more carbonic acid in order to neutralize the carbonates or the hydroxy ion. So this is the process how this respiratory mechanism is maintaining the blood pH. The pH of the initial glomerular filtrate is approximately 7.4, whereas the average urinary pH is approximately 6 due to excretion of so many non-volatile acid in the form of phosphoric acid, lactic acid, various other organic acids, so which are being produced by the metabolic process. So the pH of urine may vary from 4.5 to 8, corresponding to the cases of acidosis or alkalosis. And beyond these range, there is no scope of life, no sustenance of life. Below 6, it's very dangerous, and above 8, it is very, very dangerous condition. So it's fatal. So this ability to excrete the variable amount of acid or base make the kidneys the final defense mechanism against changes in the body pH. We will see in the subsequent slides how the kidneys are maintaining the blood pH. So this is the renal conservation of carbonates, bicarbonates, and excretion of acids occur through 4 key mechanisms. We will see one by one. The first mechanism is exchange of H plus ion for sodium ions of the tubular fluid and reabsorption of bicarbonate from the tubular fluid. So this is the tubular fluid, and this is the renal tubular cell, and this is the blood. So what is happening here? So, once there is a production of, this H plus ion in the tubular fluid, there is lot of bicarbonate. So bicarbonate has to be no uh, conserved. So it is now combined with H plus ion, and these H plus ions are coming from the renal tubular cell, and it converts up into the carbonic acid. This carbonic acid broken up into the water and carbon dioxide. So this is how there is no net loss of this carbonate in the form of this water and carbon dioxide. It retakes its entry into the renal tubular cell. Here again, there is a regeneration of carbonic acid via the carbonic anhydride, and this further converts up into H plus ions and bicarbonate. And these bicarbonates, these are very precious, so they are to be conserved. They reaches to the blood, In exchange, to no nullify the anionic effect, there's a counter sodium ion is also absorbed right from tubular fluid to renal tubular cell and finally to the blood. So this is how from the tubular fluid there is a conservation of sodium and there is a conservation of bicarbonate, right? Then excretion of H plus as H2PO4 in the urine. So we will see here, this is the tubular lumen. So what is happening here? There is a HPO4 ions. HPO4 ions, these are the phosphoric acids which are being lost through the urine, So they now combined with H plus ion, they converts up into the H2PO4 and along with the sodium, it converts up into the sodium dihydrogen phosphate, which is excreted into the urine. So H plus ions are coming from where? Like from carbon dioxide and water. There is a formation of carbonic acid and this carbonic acid broken up into H plus ions and the carbonates. And this is how there is a loss of H plus ions. So H plus ion goes here and the sodium comes in return to the renal tubular cell right from the tubular lumen and finally to the blood. So, and this counter anion is also coming to the blood. So these are the driving force for each other. For each anion, there should be a cation in order to neutralize the compartmental no ionic effect. So these are this is how there is excretion of H plus ion as H2PO4 in the urine. The next is formation of ammonia and excretion of ammonium ion into the urine. So if there is a lot of production of ammonia into the blood, that is to be excreted out. So how it is being excreted out? Ammonia into the tubular lumen, not combined with H plus ion. H plus ion can come from the from the carbonic acid via its ionization. You have seen it from the previous slide. And this H plus ion combined with ammonia form ammonium ion. And along with the counter chloride ion, in the form of ammonium chloride, it is excreted in the urine. So maintenance of pH, ion balance, and acid balance are, interrelated with each other. So this ammonia is coming from where? How it is being washed out? Basically from the blood. Ammonia is now transported in the form of glutamine. Glutamine is the major transporting form of or scavenging form of ammonia. So glutamine from the blood, it reaches to the renal tubular cell, and the renal tubular cell via the enzyme deaminase, glutaminase. So it converts up into the ammonia and glutamic acid is released. And this ammonia is now released into the tubular lumen. So this is how the ammonia is lost in order to maintain the pH and wash out the effect of alkalinity. So this is how the kidneys by various excretion and absorption mechanism, it maintains the acid-base balance.